My name is MD Rashiduzzaman. I was born in Bangladesh and I come to Germany to pursue my higher studies in the direction of optics. And then I come to Marseille for my master thesis and then continue my doctoral thesis in Institut Fresnel. There are two different kinds of cells in our immunosystem. One is called phagocytose. It eats the pathogen and then it brings back to the T lymphocyte. And when they come closer, they make a synapse. They make a interface, a specific structure. We call it immunological synapse. And from medical point of view, it is extremely important to identify, to save lives. Exactly.
Yeah, no, I think it's a good idea.
So I think if we use... In my thesis, what I do, I want to identify this synapse within very short time, for example, in 10 minutes. So how do you do it? We basically use a very well-known method. People know it from half a century. It's hologram. So we combine this hologram technique with sophisticated numerical algorithm to identify this synapse. The cell is not invisible, it's transparent. It means the contrast is not good. So presently people use some kind of chemicals to enhance the contrast. But if we use the chemicals, this is not good for the synapse. It may be destroyed. That's why the present techniques does not permit us to identify the synapse very easily. So in our case, the principle is very simple. We have a laser light and we divide the laser beam into two parts. One part directly goes to the object or the sample we want to image, and another part is just a reference. And the signal travels to the object and then it return back as a reflected signal. And when this reflected signal combined with the reference, they make a specific structure. We call it interference pattern. And this interference pattern is recorded by the camera.
Is it the best we can get?
So in between, I think this
no,
one.
Okay, you lose it.
Okay.
So
yeah,
here it's the maximum now. Okay.
Okay. Now can you go back to higher illumination angle?
To intense here. Okay, here we better see the shape of the sample here.
Okay, thank you. So far, we have successfully identified the contour of a cell. And if we have several cells, we can identify the contour of the cell. But the big challenge is, can we identify the cell and cell interface? Yes, we are now in this part and successfully very recently, we have done very good experimental data acquisition. Now we are blocked a little bit in our mathematical algorithm. So it's a dream job. Definitely it's a dream job. If we can identify it, it's a big success. So now we are very close. Because you know what? From laboratory to medical application, it's a long process. So now we are optimizing our mathematical algorithm. And finally, if we find something better, then we hope some company will invest more money on this.